group A11, and we did our presentation over the shaping of modern human immune systems by multi-regional admixture with archaic humans. Archaic humans includes Neanderthals and Denisovans, and the publication of the Neanderthal genome provided evidence that Homo sapiens bred with Neanderthals as well as other hominins, including the Denisovans. Modern humans and Neanderthals and Denisovans all shared a common ancestor in Africa long ago, and then they split, and the Neanderthal lineage headed to Europe and West Asia, while the Denisovans headed to Northern Asia. When modern humans finally emerged from Africa, they had the opportunity to once again encounter these other groups and interbreed. And interbreeding are responsible for giving us our immune system genes, the HLA, B, and C, which stands for human leukocyte antigen. They are vital to our immune systems because they respond to many disease-causing agents as well as adapt to new ones. And each gene type has hundreds of different versions or alleles. And through statistical genome-wide comparisons, studies have suggested that Neanderthals contributed 1 to 4% of genomes of non-Africans, and Denisovans contributed 4 to 6% of Melanesian genomes.
These two tables show the 3 different genes that were studied. These are the HLA genes A, B, and C. The first column is the coverage of the alleles, which is basically how much of the actual gene each one takes up. The second column right here is the actual allele differences, and the Denisovans up top and the Neanderthals in the bottom. And the last one is the single nucleotide polymorphic differences between the alleles and the Denisovans and the Neanderthals and their human counterpart that we see in modern humans today.
These tables and maps show the different allele frequencies across the world. You can see that the Africans tested, 100% of them associated with the HLA-C 1505 allele and not with any other. The African with the 2 number signs just below it is representative of African Americans. Over in the maps is a visual representation of this data. The different allele frequencies are represented through different colors, from 4% in the red down to 0 in the dark blue, and the gray areas are areas where no data was collected. This bottom table shows the putative archaic ancestry at HLA-A. These were estimated from the combined frequencies of these 6 alleles. As we noted on the previous slide, the HLA-C12 and HLA-C15, other than 1505, were not present in Africans. And this data is also a possible explanation to Native American susceptibility to European disease. And the HLA-A estimates are much higher than genome-wide estimates of introgression. And this really shows how interbreeding with archaic humans has combined with natural selection to shape the HLA system. There is also, there are many arguments against interbreeding with archaic humans. One such argument is that HLA-C15, except for 1505, is found in African Americans. 2.2% of the African Americans tested did not associate with just the 1505 allele. And the question there is really why. John Hawks has some ideas on that, and he has written about how that HLA-A11 is in Africa, but it's very rare. That's more evidence suggesting that there was not interbreeding outside of Africa. And HLA-C0702 is in sub-Saharan Africa at very low frequencies. And what this shows is that if there were any disadvantages associated with HLA-A, such as disadvantages with malaria, it shows that that's a possible product of recent evolution and that is quite possible even without interbreeding.
So to conclude our presentation, one could say that the findings of this study add to the growing body of evidence that interbreeding did happen between modern and archaic humans, thus providing possible explanation for the current immune system of humans. And these right here are our sources.